Great, thanks Tim, and thank you everybody for joining us this morning. I can see a couple of familiar names in the participant list, so this will be an update for you, some new names as well, so I'm delighted to be able to introduce AdAlta to you today. We are an ASX listed drug discovery and development company, and our mission in life is really developing high value drugs and therapeutics for diseases that have challenged traditional antibody therapeutics. So at a glance, our company is built on the back of our iBody platform. Our iBodies are a powerful drug discovery tool for creating drugs against diseases underserved by traditional antibodies, and we can use those in a modular format to create a vast variety of different formats and therapeutic modalities. We've used this platform to develop 4 programs already to date. We have wholly owned the first 2, and these are in the field of fibrosis and inflammation. Our lead product is a product called AD214, which is a first-in-class, i.e. it's the first drug using this particular mechanism of action. It's a first-in-class antifibrotic. It's completed phase 1 clinical trials successfully, and we're working towards phase 2 clinical trials with potential in multiple indications. And we have commenced discovery on a second target in the same area. So this is the first part of our business model, which involves developing drugs through to phase 1 or 2 when we out-license them to big pharma. The second half of our business model is co-development of drugs from the get-go with commercial partners. And we have 2 collaborations in the very exciting field of immuno-oncology. The first is a collaboration with Karuna Biotech to develop iBody-enabled CAR T-cell therapies. This is a $20 billion market, and we are in discovery stage on this one. And the second one is a collaboration with GE Healthcare, who's one of the leading diagnostic imaging companies in the world, and we're developing iBody-enabled PET imaging agents to address PET imaging in immuno-oncology. And then beyond that, we have the capability to build out our pipeline. We have an aspirational target of 10 programs in development by the end of 2023. But our near-term priorities are progressing the existing assets to ensure that we hit some pretty exciting near-term milestones. So another way of looking at it, and this is perhaps grounding it in what's the impact of what we do, which is the important step, of course, in being able to deliver innovation. It's something that is useful that people are prepared to pay for. So there's 4 needs that drive the value of our company that we're trying to solve. The first one is that antibody therapeutics have been revolutionary for the pharma industry over the last 30 years, but they still can't do everything. And there are a significant number of drug targets they've been unable to successfully address. And our iBodies are a differentiated antibody-like platform that can potentially address many of these diseases. And for this reason, pharma companies look to us to provide solutions to some of their more challenging drug discovery problems. If you then look at the assets that we're developing, our AD214 for fibrotic diseases, a good example is idiopathic pulmonary fibrosis. This is a degenerative and fatal disease affecting a significant, albeit small number of patients around the world. It's a $3 billion market today, driven by 2 drugs that don't work particularly well. And there's a desperate need for new approaches to this debilitating disease. AD214 could be one of those. In the field of CAR T-cell therapy, these therapies where we're genetically engineering your immune system to fight cancer have revolutionized outcomes for cancer patients, or at least for blood cancer patients. What we aim to do and what the world needs is now to extend that same hope to patients with solid tumors, and our CAR T collaboration with Karuna Biotech is designed to do just that. And then finally, if you look at the field of immuno-oncology drugs generally, these are molecules that take the brakes off your immune system to allow them to fight cancer. They've revolutionized outcomes because we now talk about long-term cures, but they only work for 20 to 40% of patients. And so the opportunity, the need is to identify who those patients are as soon as possible. And we can potentially identify those patients through our imaging program with GE that will enable us to identify those responders much earlier and get them onto the right therapy much faster. So they're the kinds of unmet needs and the key sort of focus areas that AdAlta is working on today. So let me step through each of those 4 areas in turn. Let me touch on our eye bodies first. So these are what we call single domain antibody-like molecules. These are very small format antibodies, and we mimic the structure and features of a natural single domain antibody found in the shark immune system. But we do this using a human protein that we found maps onto exactly the shark scaffold or backbone. We engineer the binding domains into that human protein to create a mimetic of the shark single domain antibody system. This is a really small protein. It's 10% the size of a traditional monoclonal antibody, and it's half the size of the fragments normally used in a CAR T cell therapy. So what are the advantages of these relative to a traditional antibody? Firstly, we confer all the specificity and selectivity of an antibody versus a small molecule. But what we can also do is the small size and the long binding loop mean that we can access drug targets that are much more difficult for the big, bulky antibody to address. And this includes receptors that penetrate the cell membrane and are influential in telling a cell how to respond to the environment. So that diversity gives us the ability to do things that antibodies can't. The small size also means we can use this as a module in a variety of therapeutic constructs. So we can use the native or naked eye body as a therapeutic. We can use a number of techniques to extend its half-life or its duration of residence in the blood, which can be important for enhancing therapeutic effect. And 8214, for example, is an example of what we call an FC fusion protein that's designed to do that for fibrotic diseases. We could couple 2 eye bodies together to create bispecifics. We can attach a therapeutic payload to the eye body, where the eye body now is being the delivery vehicle to deliver the therapeutic modality to the right place. And this is what we're doing with PET imaging and with CAR T cell therapy. So, you know, really powerful, really diverse drug discovery platform that provides unique capabilities inaccessible by traditional antibodies. So let me now turn to 8214. And specifically, our lead indication for some time, but not the only indication, has been a disease called idiopathic pulmonary fibrosis. This has involved scarring of the lungs, which results in them progressively and irreversibly getting stiffer and less flexible. And you can imagine from the impact of a scar on your skin, if that's happening in your lungs, breathing becomes incredibly difficult. There are over 300,000 people living with IPF around the world today. They have a duration of survival less than 4 years from diagnosis on average. And the 2 current therapies that generate $3 billion in sales a year are the best we have, but they have really limited effectiveness and some pretty serious side effects. And many patients can't tolerate these drugs for more than a year. And further, with the increasing burden of COVID-19 and long COVID, we're expecting that this is going to increase the amount of fibrosis in lungs and other organs, increasing the need for antifibrotic drugs. And this was flagged as early as the first couple of months of the pandemic in 2020. So this gives you a flavour of the debilitating disease we're wrestling with. Now, what we've done with 8214 is complete a usual and extensive campaign of preclinical development. We've optimised the molecule to produce 8214, which is, as I said, a first-in-class antifibrotic. First in class because we're the only drug targeting a receptor called CXCR4 for fibrotic diseases. We've also completed a phase 1 intravenous clinical study demonstrating that 8214 was well tolerated and that it binds tightly to CXCR4. And we have extensive preclinical data supporting efficacy of the IV administration of this drug. We completed the phase 1 study in the middle of last year. We booked the drug material manufacturing for the next clinical trials at that time. COVID has driven out supply chains for biologics of all kinds, and we won't get that material until late next year now. And so the next clinical studies are not due to commence until the first half of 2024. Now, that was a little disappointing for us, but what it actually did was create some significant options for us to actually optimize the value of this product as we head into the next clinical trials. We know that this has multi-indication, and now we know it's also got multiple partner potential. There are a number of large pharmaceutical companies we've been in discussions with for some time. They all have different interests in terms of the indication that they would prefer. Some are lung specialists, some are kidney specialists, some are eye specialists. And we started to think about, well, how do we partner this drug across all indications? And so what we have at the moment is the opportunity to develop unique indication-specific formulations for AD214 that enable us to then achieve that indication-specific partnering and ultimately potentially partner this drug 3 ways rather than 1 way. And so as a result, we're now deep in preclinical development of an inhaled formulation for IPF or lung fibrosis. That will enable us to then have inhaled, if successful, have inhaled delivery for lung fibrosis, intravenous delivery for kidney fibrosis, and intravitreal or direct eye delivery for eye fibrosis. And we're aiming to finalise the routes of administration that are available to us and the indication priorities for our clinical program in the second half of 2022, based on a significant amount of data that's emerging in the next 3 months. Why inhaled for AD214? Firstly, it improves bioavailability. We know that AD214 happens to clear pretty quickly through the liver, and so we avoid this by direct lung delivery, which reduces the amount of drug required. It also gives us greater flexibility for optimizing dosage. Inhaled is going to be way more convenient for a patient. We're using a handheld nebulizer device. This is much more convenient than having to go into the clinic every couple of weeks for an infusion. And it enhances cost effectiveness, not just because we're using lower drugs, but because the total health system costs to administer the drug become lower. So this is a way superior formulation to intravenous. And what this enforced delay driven by our manufacturing process has done is enable us to pivot not just from the race to the clinic, but now to how do we maximize the options we have available for this drug, both from a partnering and a clinical impact perspective. So the milestones coming in the next 6 months or so. During this quarter, we'll be able to demonstrate the antifibrotic effects of AD214 in cultured human lung tissue. We'll have imaging, distribution, and retention of AD214 in sheep lungs, which are the best animal model of human lungs. And this is showing and confirming the simulations we've already done to show that we can get AD214 into the far reaches of the lungs. And we're studying efficacy in a bleomycin mouse model of fibrosis, which is the gold standard model of efficacy, all via inhalation. And these studies are anticipated to provide clarity around toxicology strategies and manufacturing strategies, and also provide and accelerate our existing partnering discussions for this asset. All of those studies that are reading out in September are underway. We've completed pilot imaging studies in the sheep showing that we can get AD214 distributed to the far reaches of the lungs. And the mouse study is now into the analysis of the results stage. So we've completed all the treatment of the animals there. And this can be a really valuable asset. These show some recent deals in IPF, noting that some of the acquirers, Roche and Boehringer, for example, are the marketers of the existing drugs. They know there's a need for better outcomes. Even at phase 1, these assets can command significant milestones, a 45 million euro upfront for the BridgeBio asset at phase 1. And this is 3 times our current market capitalization, just to give you a sense of the impact that a transaction could have on our company. So let's quickly turn to the immuno-oncology assets. Let's talk about CAR T-cell therapies very briefly. These are revolutionizing or a new approach to treating cancer, because what we're now doing is reprogramming a patient's own immune systems to be able to see cancer as a pathogen. CAR T-cell therapies stands for chimeric antigen receptor. We've got an artificial receptor or targeting agent on the T cells. Essentially provides a living drug that is administered once and then can continue to treat and attack cancer as it continues to come back over time. The first products were approved in 2017. There are now 6 approved for blood cancers in the US. The market is forecast to reach 20 billion by 2028. And by 2030, more than half of those revenues are expected to come from solid tumor CAR T cells. So what's the benefit of using an iBody? So the small size means that there's actually much more room in what we call the gene cassette or the vector, which is used to insert the CAR receptor. So this is a model of the CAR receptor. Essentially, the iBody on the outside targets a molecule on the cancer that's specific to the cancer, And once bound, it then triggers an activating cascade that tells the T cell to go and kill. Now, we can only fit so much size into this, into the vector that delivers these CARs. And so having a small size means you've got greater probability of putting superior functions and increases the ability to add a dual targeting CAR, for example, which is really important to treating solid tumours. The long binding loop, again, enables us to access tumour antigens or molecules on the surface of the cancer that otherwise wouldn't be possible. And we've got greater control of the interaction between the T cell and the cancer, which is really important to be able to fine tune that to optimize the effectiveness of the CAR T cell therapy. So all of this, we believe, leads us to be able to develop superior iBody-enabled CAR products, generating CARs against novel tumor antigens, generating dual and bispecific CARs that enhance specificity and reduce tumor escape, and potentially even secrete small antibodies that can help enhance the longevity of the CAR T cells by reducing the tumors, the immune suppression that a tumor induces. So we have an existing collaboration with Carina Biotech to create iBody-enabled CAR Ts against up to 5 different targets. We have successfully generated CAR T cells with Carina and demonstrated in vitro cell killing in our against our first target. And we are expecting further data from that collaboration in the next 6 months. In terms of PET imaging, again, the immuno-oncology market, as I mentioned, is about taking the brakes off cancer or the immune system that cancer imposes. And what we know is that there is an enzyme called granzyme B that's produced by immune cells. And there's very good data now to show that if a patient is going to respond to an immuno-oncology drug, then we'll see an increase in granzyme B in the vicinity of the tumor. It tells us that the T cells are killing. And so if we can image that granzyme B, then we'll be able to work out which of those patients are responding to those therapies much earlier. GE came to us and said, can you find an iBody against granzyme B? If so, will it use our radioisotopes to create the imaging agent and potentially access a market that, today for the total PET imaging agent market is worth about $6 billion. The largest products are about 400 million. The attractiveness of this market, although the royalties and milestones will be smaller than for a therapeutic, is we get to market much faster. So even single-digit royalties in a $400 million market become a really interesting number for us and could be, not more than 3 or 4 years away. So this collaboration was fully funded by GE through the early discovery phases. We have developed a panel of iBodies, preclinical proof of concept studies, and further iBody optimization are underway, and manufacturing development is also progressing with GE. Milestones across our immuno-oncology pipeline are set out here. During this quarter, we expect to select the next two targets with Carina, but also have more detailed and more extensive in vitro cancer cell killing assays for our initial target. We expect to have full in vitro cell killing assays complete by the end of the year, which will enable us to choose the indication for in vivo proof of concept studies. We're updating on GE in consultation with GE and as milestones are achieved. Back to the company as a whole, current market capitalization of around about 15 million, a very loyal and supportive shareholder register, which we're very appreciative of, and $8.7 million of cash at the 30th of June, which with the ability to flexibly defer our manufacturing toxicology campaigns gives us plenty of runway through the milestones in the next 6 months. And we're burning around about just under 2 million a quarter. So as an investment proposition, we think we have a really powerful drug discovery tool that is in demand from pharmaceutical companies. We have 4 very exciting programs under development already, the lead of which is approaching to phase 2 and ramping up partnering discussions. The 2 immuno-oncology collaborations are addressing significant markets and are progressing well through discovery. We have a team capable of leveraging the platform aggressively, and they're leading experts in discovery. We have a clear vision for how we progress both the existing assets and grow the pipeline, and some really exciting news flow in the next 6 months, or 3 to 6 months, that we think will be demonstration of the significant progress we're making with this very exciting platform. And so with that, Tim, I'll stop and see if there are any questions.